Practical, understanding the functioning of a tabletop high-speed centrifuge. Aim, to understand the working principle, operation, and functioning of a tabletop high-speed centrifuge, including setting parameters such as speed, RPM/RCF, time, and temperature for efficient sample separation. Materials required. Equipment. Tabletop high-speed centrifuge, example Eppendorf centrifuge. Rotor, fixed angle or swing out type. Centrifuge tubes, microcentrifuge tubes, 1.5 to 2 ml. Samples. Dummy samples, water or buffer solution, or biological samples. Accessories. Micropipettes and tips. Tube rack. Balance, for proper tube balancing. Ice, if temperature-sensitive samples are used. Principle. A centrifuge works on the principle of centrifugal force, which separates components based on their density, size, shape. When samples are spun at high speed, denser particles move outward and form a pellet. Lighter components remain in the supernatant. The force applied is expressed as RPM, revolutions per minute. RCF, relative centrifugal force, times G. RCF is more accurate and depends on rotor radius. RCF equals 1.118 times 10 to 5 times R times RPM, 2. Method/procedure. Step 1, instrument familiarization. Observe the centrifuge components. Lid with safety lock. Rotor and rotor slots. Control panel, RPM/RCF, time, temperature. Step 2, sample preparation. Fill centrifuge tubes with equal volumes of liquid. Ensure tubes are tightly capped. Step 3, balancing the tubes. Place tubes opposite each other in the rotor. Ensure equal weight to avoid vibration and damage. Step 4, loading the centrifuge. Insert tubes properly into rotor slots. Close the lid securely. Step 5, setting parameters. Set required. Speed, example 10,000 to 15,000 RPM. Time, example 5 to 10 minutes. Temperature, if refrigerated, example 4 degrees Celsius. Step 6, running the centrifuge. Start the centrifuge. Observe acceleration and smooth operation. Do not open the lid during operation. Step 7, completion. Wait until rotor stops completely. Open lid and remove tubes carefully. Observe pellet formation. Results. The centrifuge operates smoothly when tubes are properly balanced. Separation of sample components is observed as pellet at the bottom of tube, supernatant above it. Speed, time, and temperature directly influence separation efficiency. Conclusion. The tabletop high-speed centrifuge effectively separates components based on centrifugal force. Proper operation involves correct balancing, appropriate selection of speed, RPM/RCF, safe handling. Understanding its functioning is essential for accurate experimental results in biological and biochemical laboratories. Precautions. Always balance tubes properly. Do not exceed recommended speed limits. Never open lid while spinning. Use compatible tubes for high-speed operation. Maintain temperature for sensitive samples. Applications. DNA/RNA extraction. Protein purification. Cell/organelle separation. Clinical diagnostics.